Hello, this is Dr. Paul Cottrell. I'm going to be talking about this paper that is called Uncanny Similarities of Unique Inserts in the 2019 Novel Coronavirus Spike Protein to the HIV-1 Glycoprotein 120 and the GAG protein. Okay, so just to give you a little background, I read the paper and I did some analysis. Now, what this paper focuses on is kind of two things. One is the similarities of the Wuhan strand to SARS coronavirus GZ02, and the ID number is AY390556. When I did my blast analysis a while back, about 5 days ago, I had the theory that it was probably coming from bat SARS and derivatives of bat SARS, and there was some sort of bioengineering copy paste into the Wuhan strand. Now, their analysis focused on the front half of the protein, the S protein. All right, and I was focused on what was missing from bat SARS to the Wuhan strand. So I was focused on more of the big gaps. And they have done an analysis where they're stating that the Wuhan strand is more similar to this GZ02 strand. And I was, that number has less homology when you look at the total genome than the bat SARS. So I was looking at the whole genome and the whole S protein. So I still think that the total homology is still bat SARS. You may have some areas, very small areas, that have closer homology to a different strand. Okay, so that's kind of the difference between what I did in the blast analysis and what this paper is saying. But the bottom line is that Wuhan came from a different strand of SARS, and they didn't focus on the big chunks that have been copied into the genome. They focused on the area that seems to be zoonotic, or maybe naturally selected in the lab. The reason why I'm saying that is that if we go to the next part of the paper, they start to compare Wuhan to HIV, the glycoprotein 120, and the GAG protein. Now, glycoprotein 120 is from an area of the genome for HIV that codes for a protein that's actually GP160, and then GP160 is broken down into GP120. So this amino acid was compared to the amino acid sequence of the S protein, and they found that this S protein is about 1,200 or so amino acids, and I believe that's right. Let me just double check here. The S protein is N. I'm tired. It is. Is it? I don't know why I'm killing myself for this. How long is this thing? That's the protein ID, gene ID, protein ID. Okay, it is 1,273 amino acids. So their paper compared the kind of like the front end of the region, maybe halfway too. So they had a small section that matched HIV, glycoprotein 120, near the 71 range, and then they found another small sequence that matched, now these are amino acid sequences, that matched 145. And it's important. I'll explain why. It's a small sequence. It's almost like six amino acids or so around the 145 area of the Wuhan amino acid chain for the S protein. Another match at 245, and then they looked at HIV gag and saw that there was a similarity, but I don't think it's that strong, the similarity, because the gag protein for HIV is longer, and there's only the tail ends of this that kind of match, and it's not a perfect match. So there is some similarity, and those similarities are 1, 2, 3, 4, 5, 6, 7, 8 amino acids that match, 4 on each side, okay? But it's a longer chain, while the others were more of a, 1-to-1 match, except the 245 region where there was a gap in the S protein and not a gap, relative to the GP 120. Now, what is GP 120? Like I said, it comes from GP 160. It's cleaved, and this helps HIV to go into the T cells. It's the CD4 plus cells. So it's very similar concept as the S protein, where it's a spike from the capsid, and it signals when it attaches to a cell to go in. Okay, it's kind of like the key. Now, 120. These proteins are classified based on size. So this is kilodaltons. So it's 120 kilodaltons. Kilodaltons. So the gag is very similar to the. I did a video on the open reading frame for 3B. I think it was 3B, 3B by this Piang Zhao. He did this research paper back in 2015 or something like that, where he took bat SARS and seemed to be playing around with the open reading frame for 3B, which is the protein, the capsid protein, that helps to assemble the protein. Well, that is very similar to GAG for HIV. So the open reading frame 3B is the capsid assembly for SARS or coronaviruses, and GAG is the assembly protein for HIV's virus. Now, what is interesting to me is these little sections. See, amino acids sometimes are polar or non-polar, okay, depending on how many nitrogens they might have, because amino acids have a different structure, right? And as they're building up, you can have charges, and if they're polar or non-polar. So a polar region is going to be usually outside of the protein, facing outside, while a non-polar region would be hydrophobic and it will be inside the protein. So by having these charges, it can help create these structures. So these changes are affecting it looks like it's affecting the charges of certain regions to allow for it to be more more adaptive to get the cell to accept the virus. Okay, so in the membrane, there's lipids. All right, the polar charge of the lipid is facing either outside or inside the cell. The tails of the lipid is what's the inner part of the membrane. Okay, so things that are non-polar, like certain steroids, will be able to go through the membrane. But but water molecules have a harder time. Polar stuff will kind of stay out. Okay, so by changing the charge of this protein, this S protein, that has kind of like a charge homology to AIDS or to HIV, it will probably have a more affinity. I suspect that I think that what they were doing was that they created Wuhan, bioengineered Wuhan, and was trying to increase the affinity of the S protein to bypass the immune system for research, because we know that they were trying to use this virus to study latency and how it can be in an organism for a long time, let's say a bat or swine or whatever, and they were investigating how that might happen, and then maybe create a vaccine against a very strong strand of this. And in the zoonotic world, the theory would be that the vaccine would be able to fight against the weaker strands because they designed a vaccine on a very strong strand. That's my working hypothesis on this, but it's for sure, in my humble opinion, that Wuhan strand was bioengineered by bat SARS and its derivatives. But what's interesting is that there is this little tweak that was going on outside of the big chunk that was copied in. It's left of that. It's a little bit, it's a little bit left in the genome. just adjacent to this copied area. And, again, the genome, as it's coding, each of the three nucleotides will code for a protein. And there's redundancy. So if there's a nucleotide that happens to mutate, the probability of changing the amino acid is reduced because of redundancy. But if enough changes take place, then the amino acid will change, or create actually a start or stop codon. All right. So the chances of randomness, of having the GP120 homology and the GAG homology by accident, just by natural selection within a zoonotic world, I guess maybe it is possible, because you're only talking little chunks of the actual GAG protein or the GP120. But it is suspicious. Now, there's a statement in here. And I'm just surprised that in the paper I didn't see anything that was talking about the big copy paste from bad SARS to the Wuhan. All they focused on was that adjacent sequence. That's the front end of the amino acid, front end of the protein for S protein. But this uncanny similarity of novel inserts. Well, the novel inserts, the novel inserts they only focused on was related to the GZ01 or 2 strand and the HIV. But these novel inserts in the 2019 novel coronavirus spike protein to HIV-1 and the glycoprotein 120 and the GAG is unlikely to be fortuitous. So, they're basically saying that there's a low probability here that this is just by chance. Now, so I agree with him on that. I'm just surprised they didn't focus on the copy paste. That I don't. So I think the working hypothesis right now, based on this paper and the things that I've been doing in BLAST for the NIH, I believe it was bat SARS, possibly that MK strand, and that area of the S protein was copied in. There was a big chunk that was copied in. Not all of it was copied in. There was something else added that's missing from the normal homology of the evolution of this coronavirus family. All right. There was stuff copied for the replicase that was put in, which this paper does not address the replicase at all. That seems, to be somewhat bioengineered. But it's very interesting in how they kind of focused on, well, there's a tweak in the non-copied region that seems to have homologies of sections of protein, of 2 proteins from HIV. That is focused on the attachment mechanism of the coronavirus. All right. Now, most of the inserts that they're talking about from the HIV are from the GP120, which is HIV's attachment to CD4 plus cells, immune cells. That's why AIDS was so hard to understand, was because it was suppressing the immune system, and lots of other diseases happen when you suppress the immune system. People that have cancer and their immune system is down, they because of chemotherapy or radiation therapy, a lot of infections can arise. So that's why it starts as a syndrome, because they don't really fully understand what's happening. And the gag protein for AIDS, for HIV, is for the assembly of the protein, of the assembly of the virus. So that being part of the S protein to increase its affinity to cells is a little strange to me. It's possible that this was somehow naturally selected in the lab. I have a hard time believing that they knew where to copy paste these little sections in the genome to create the right sequence, the right folding of the protein for it to match. It makes a lot of sense to me that they did these big chunks of copy paste into the genome. All right. But, and that's a much easier thing to do, but to tweak it, because you're talking about, remember I said six for some regions, like the first part of the insert is 6 amino acids. Well, that means there's 18, because there's three nucleotides per amino acid. So it's 6 times 3. So 18 continuous nucleotides had to be inserted in at the right spot for this to fold correctly. And the way this is working is that it is folding these inserts aren't contiguous. So it's folding in such a way that it allows for it to have the right properties of charge to be able to have more affinity to the cell. All right. So in the lab, you can select for that and keep on looking for variants as they divide and mutate for the proper selection. All right, and then it's like, okay, that is the strand. It has a strong affinity to these types of cells. All right, now we isolate that virus and grow it, and now we have a batch that has a strong affinity towards a certain receptor. All right, that to me makes more sense. Their statement doesn't necessarily mean that the HIV sections were put in by CRISPR or put in by some sort of restrictive enzyme. All right. It could have been selected in the lab. It's a possibility that there was a natural selection within the lab to create this S protein on top of the actual insertion, human insertion of genetic code in the other region of the S protein and in the front end of the region of the replicase. So I think maybe two things were happening here. There was bioengineering and then kind of like a tweak of selectivity for CD4, CD4 plus cells, and had affinity to lung tissue, let's just say, endothelial tissue. This makes sense to me, that it wasn't by chance, but I think there were two wasn't by chance paths that it took. Bioengineering for part of it, and natural selectivity for part of it within a lab environment, not zoonotic, in the wild. That's my working hypothesis. But what is interesting is that the clinical evidence of the patients in China are showing problems with white blood cell count. And if this virus can attack your leukocytes, then you're going to have this, you're going to have this downregulate. You're going to have less white blood cells, and you're not going to be able to fight the infection, which is probably what they were trying to select for, so that it would stay more latent in the body. Because if you had an immune system that was fighting the virus in the organism, then it wouldn't stay in the organism for as long, because it would either be cleared by the immune system too quickly, right? So I think they were selecting viruses in the lab for it to be latent. And this is part of the reason why we're seeing these, this long latency that's taking place. And I think that the potentiality of this affecting the immune system is very high now. Now, I did a video where I think one possibility is that something's happening with PD-1 and it's being downregulated. Now, it might be possible that this S protein, this new selected S protein with the HIV domains, or a similar homology from GAG and GP120, might signal immune cells to downregulate PD-1. And if that's the case, then that is the mechanism that I was looking for, that actually would say, kill the working antigen presenting T cells and upregulate suppressor cells, T suppressor cells, or at least keep them constant and downregulate the T cells. Because normally when you have an infection, your white blood cell count should go up, not down. But in at least what has been leaked out is that people are having white blood cell counts that are going down. So maybe this new thing that is in the S protein is actually affecting PD-1, and that using like a PD-1 inhibitor might help with preventing this thing, this S protein, from bringing up, from preventing. By having a PD-1 inhibitor, it might actually bring up your good T cell count. But I don't know, because I don't have the cells to say, yeah, that's what's happening. I mean, but we have heard cases where individuals are being treated with AIDS type medication Well, that screams to me that this is affecting CD4 plus cells and doing something to the immune system. And maybe it's the PD-1 pathway, maybe it's another pathway. I don't know. And I'm, as I dive into this a little bit more, maybe I'll figure it out. But definitely this thing, I think, it stays latent because it somehow can manipulate and downregulate the immune system. And it's very possible that it may stay kind of dormant within an infected cell in the lung, and then something happens in the immune system that signals these infected cells to actually start assembly of the protein. So there might be some dual thing going on where the virus goes into the cell, stays dormant. Some of the other viruses that are floating around disactivate the immune system. Something happens in the immune system where it gets reactivated, realizes that there's cells that are infected, and then basically starts to try to kill those cells. And as that's happening, it creates a cascade that activates the production of the virus to get kicked out. That's a working hypothesis. But that's my take on the paper. I find that it was very interesting and very helpful. It helps explain what was going on with the lower. It helps point in the right direction on why we're starting to see lower white blood cell counts. I disagree with their methodology on just focusing on the front part of the genome for the S protein. They should have looked at the whole thing and what the homologies were, so they can show that a big chunk of that was bat SARS. And I was very surprised that they just focused on the S protein and they didn't say anything about the replicase, because there's two big sections that have been copy pasted in, and it's very strange, very strange. So that's my take on this, and hopefully you learn something, and maybe someone has another idea, because the PD-1 idea was someone reached out to me and said, hey, there's some talk about using PD-1 inhibitors for some of these patients, and that kind of like, oh, why, but here we go. This is what I mean by it's important for the people that are listening. If you're a nurse or a doctor, please reach out to me. I'd love to talk to you about what you think is going on. Even if you're retired, you have a wealth of knowledge, and you can help us understand what's going on here. But if you're a doctor or a nurse, that especially the nurses, I think, are the front line of this whole thing, and they probably know more than anybody. I would love to hear from you, because I want to understand what you think it is, and what you think probably the right clinical path should be for a good outcome. But if you know someone that is a nurse or a doctor, an MD, that is not watching, please share the videos with them and explain, hey, there's some crazy guy out there on the internet that's trying to figure this out, and maybe you can help, or maybe there's some ideas. But well, thank you for listening, and have a nice day.